Hello, hello, hello. Good afternoon, good evening, good morning, everyone, depending on where you are joining us in the world. Thank you for joining us for yet another edition of the GSU Malaria General Club monthly series. Welcome to all of you. Today's webinar is a vector-focused one in honor of World Mosquito Day, hashtag World Mosquito Day, which took place on the 20th of August, which is an annual event, which is a necessary commemoration of the British doctor Sir Ronald Ross's discovery in 1897 that female Anopheles mosquitoes transmit malaria between humans. And this general club will be referencing Dr. Kelly Bennet's paper, The Anopheles coluzzii range extends into Kenya, detection, insecticide-resistant profiles, and population genetic structure in relation to conspecific populations in West and Central Africa. I hope you all followed Malaria Gen's channels on LinkedIn and Twitter for World Mosquito Day posts. Please do go to those channels and catch up if you didn't, because there was a lot of resources that were shared leading up to and on World Mosquito Day. My name is Pablo Chauke, for those of you that do not know me, and I'm a training coordinator at Malaria Gen, working with a team of awesome people, some of them who are in the room right now, to organize these monthly events. Just a few ground rules. The General Club is being recorded and will be available on a Malaria Gen YouTube channel later, so you can share it with your friends and colleagues in your networks later on in the day. Please do check out the Malaria Gen YouTube channel for this recording of this webinar. Kindly stay on mute so that we don't disturb the speaker. We will have a Q&A session at the end where you can raise your hand or type your question in the comments in the Q&A box or the chat box as well. Without further ado, I would like to introduce our speaker for today, Dr. Kelly Bennet, a hardworking and kind colleague. We've been working together for a few years now, and she kindly agreed to give this webinar when I invited her. So thank you for accepting the invite.
Thanks so
The
much for
title of the talk is going to be similar to the paper. I'm just going to read a quick short bio for Dr. Kelly Bennet so that you have a clue of the type of person she is and the work that she does. Dr. Kelly is an evolutionary biologist interested in disease vectors. Her research first focused on the population structure and demography of African Aedes mosquitoes, as well as their invasive expansion. More recently, Kelly's interest has been understanding the ecology and adaptation of disease vectors across environmental landscapes and in response to biological competitors. Since joining the vector surveillance team at the GSU, she has been working to accelerate the genomic surveillance of Anopheles mosquitoes for disease control. Kelly, thank you so much again for joining us. You may begin your presentation.
Thank you so much. I'll just go ahead and share my screen. So first of all, I just wanted to thank Pablo and Shri as well for organising this. I'm really happy to be given this journal club. And today I'm going to be talking to you about some work that I have done to investigate the taxonomic status of Anopheles mosquitoes in Kenya. And this work was headed up by Luna Kamau at the Kenya Medical Research Institute in Nairobi. Unfortunately, she couldn't make it today, and so I'm standing in her place. So today, I don't expect the presentation will take up the full allotted time, so you might get back some of your day, but we'll see where we get to. So of course, malaria is responsible for a high amount of mortality and morbidity worldwide and remains a very significant health concern in Kenya. So currently, about 70% of the population are at risk, and there are about 3.4 million cases and 12,000 deaths every year. There has been significant improvements in these numbers in recent years due to efforts to roll out insecticide-treated bed nets, increase indoor residual spraying, and also to improve preventative treatments and diagnostics as well. So these cases were, in fact, above 5 million just a couple of years ago, and so they are reducing, but obviously there is still significant work to be done. In Kenya, the worst impacted regions are these regions on the map that are shown in red. So we have the area here, which is the lake endemic zone. This is the worst affected area in Kenya, and most of the efforts to reduce the malaria burden have been targeted here. But there is also very high malaria burden on the coast as well. So the major malaria vectors belong mostly to the Anopheles gambiae complex. Anopheles fenestus is also a malaria vector of concern, but is not part of this species complex. The species complex is a group of mosquitoes which are morphologically indistinct. So we cannot tell which species it is based on an entomological key. And so we need some information to do with their genetics to be able to designate them as a species. And they're designated as a species because we have evidence that there is restricted gene flow or divergence based on the genome. There was a time when the complex only had 2 members in it, and these are the species that we know today as Anopheles coluzzii and Anopheles gambiae. But we now know that there are over 10 morphologically indistinct species in this group. The earlier studies were designating these based on cytogenic studies that showed there were different chromosomal forms, and also that there were reproductive barriers on crossing the 2 forms. But today now we have whole genome sequences, and so we are actually able to pick up more morphologically indistinct species. So the different species that are within the complex are typically identified based on just a restricted set of markers. They're usually typed using PCR, which targets specific nucleotide differences, either on regions of the ribosomal DNA, such as the intergenic spacer IGS, or perhaps this region called a short interspersed nuclear element, which is present on the X chromosome. So this is basically a PCR that is targeting single marker variation that is thought to be fixed in the different species. But species-specific diagnostic techniques may be used if you're expecting a particular species, and so you might actually not pick up a species that is present if you are using primers that just target this particular species. But also PCR may fail because of variation, for example, at primer sites or poor DNA quality, but it can also just provide false negative results. So identification based on just a few markers is perhaps not the best way to identify a species. However, most of the information we have about the Anopheles gambiae complex to date, for example, their geographical distributions and also ecological information, is based on these diagnostic methods that are just targeting just a few markers. And we also don't have very comprehensive sampling across Africa as well. So there's a lot to learn about where and when we find different Anopheles species. So this is what we currently know about the distributions of the major malaria vectors within this complex. So these maps are taken from the Malaria Atlas website, and it's sort of based on where we expect to find them, based on records of their presence. And the areas in red are the areas where we expect these species to be found most commonly. So we have a map for Anopheles coluzzii, a map for Anopheles gambiae, and also a map for Anopheles arabiensis. If we compare these, we can see that the distribution of Anopheles coluzzii is restricted compared to the other species. We actually only get them very commonly in sort of West Africa, and then moving across into Central Africa, across this sort of northern region here. If we look at Anopheles gambiae, we can see the range extends much further east and into Kenya as well, and a little bit further south. And then when we look at Anopheles arabiensis, this goes even further south, and they're excluded from this sort of lowland tropical rainforest area, and they extend further north. So what we can see is that we don't have a record of Anopheles coluzzii from Kenya currently. However, we do have Anopheles gambiae and Anopheles arabiensis and these are considered the major malaria vectors in Kenya at the moment. So the differences in these species ranges are due to differences in their ecology. So it's common to find Anopheles gambiae in quite humid environments, and in fact they seem to thrive under these conditions. And they lay their eggs and develop in temporary water bodies. So for example, puddles or channels that have arrived due to abundant rains. And due to this, you normally find them to be very abundant during the rainy season. Anopheles coluzzii, in comparison, have a higher resistance to arid conditions. And when you compare to Anopheles gambiae, for that reason appear to be more abundant in the dry season, but it's also because they tend to develop and lay their eggs in more permanent water bodies. So for example, fish ponds or areas of rice irrigation, for example. And they can live in these environments because they have adapted to survive and avoid predation in these water bodies. Perhaps as a result, or the other way around, they are also more tolerant of human-modified environments. So for example, environments that are affected by pollution or with higher levels of salinity. Anopheles arabiensis, like Anopheles coluzzii, thrives in arid conditions, but it's more of an ecological generalist, so it can actually either develop in temporary water bodies or permanent freshwater bodies. And then a major difference between this species and Anopheles gambiae and Anopheles coluzzii is that it rests and feeds outside, whereas the other two species, they're more anthropophilic and bite indoors. So the ecological differences that you see between species is one reason why it's so important to accurately identify the species that you have in a particular geographical region. And that's because differences in life history determine when, where, and how likely a mosquito is to transmit malaria. Knowing which species are present informs on malaria risk over space and time. Different species are also differentially impacted by intervention strategies that are commonly deployed. So, for example, one example is Anopheles arabiensis in comparison to Anopheles gambiae and Anopheles coluzzii. Because this is an outdoor biting mosquito, it may not be as impacted, for example, by indoor residual spraying. But different species also have different insecticide resistance mechanisms. So even in the same place, exposed to the same insecticide, different species can have differences in their standing genetic variation, which means that they have a different molecular response to insecticide pressures. And an understanding of this is, of course, important when considering which intervention strategy is going to be most effective in a given area. So for our study, we wanted to use whole genome sequencing to investigate the taxonomic status of mosquitoes in Kenya to get a better understanding of the species distributions across the country. We unexpectedly found Anopheles coluzzii for the first time in Kenya, and so we sought to look more deeply into their population structure and to understand how those are connected in relation to other populations across Africa. And then we also looked at their molecular insecticide resistance profile, and we wanted to see whether this matches up with other populations across the country. And of course, this is also very useful information for public health. So here is a map showing our sampling. It was done across 5 locations in Kenya. So we had Teso, which is in the Lake endemic region that has a very high prevalence of malaria. We have Turkana in the northern part of the country. This is a very arid region that has seasonal malaria transmission. Then we also had 2 locations in the center, which are highland regions that have seasonal malaria transmission. And then we also had samples from the coast, where there is also high malaria prevalence. So these were collected as larvae across multiple breeding sites, and over 1,000 mosquitoes were collected. And then of these, some of these went for sequencing, and in total, passing all quality controls and successfully going through all the data processing that we do, we had 564 mosquitoes in total. What I have here is a table that shows all of the mosquitoes that we collected and how they were identified. So this is based on a couple of identification techniques that I will talk through on the next slides. But basically you can see that we have Anopheles arabiensis, then unexpectedly Anopheles coluzzii, Anopheles gambi, and Anopheles quadriannulatus. So it seems that Anopheles arabiensis are very widespread across the country, as we have these from all of our sampling sites. Anopheles coluzzii was just from Turkana in this northern region up here. You can see that this is from 2 different years and across different months. So this isn't just a freak sampling event. This was sampled multiple times, showing that it is stable in this region. We have gambi from just the central region, and then we had a few quadriannulatus from the coast. So the finding of Anopheles coluzzii was unexpected, given that they hadn't been found previously, and it could be that this was sort of limited by inaccurate species identification, or that it's simply been missed. And so I'll talk through how we came to this conclusion that they were present in Kenya. So we used a couple of different methods to investigate taxonomic status. And the first thing we did was to look at ancestry informative markers. So these are SNPs or polymorphisms that are exclusive to each taxonomic group. So that means that they can be used to distinguish the different taxa. There are 2 sets of SNPs that we use. The first is a set of 2,500 SNPs that are able to distinguish the closely related sister species Anopheles gambi and Anopheles coluzzii from Anopheles arabiensis. And then we get a smaller set of 700 SNPs, which is then able to differentiate Anopheles coluzzii from Anopheles gambi. So here we have a plot of aims. What we have is that the different rows are individuals, and the columns are a SNP or variant. And they are coloured according to what we expect it to be if it is, for example, Anopheles coluzzii or Anopheles gambi. So at the top, we have our 26 individuals that were classed as Anopheles coluzzii. They're coloured in red. And then below we have Anopheles gambi. So that you can see that across the genome, we have pretty much all of the sites are coloured according to the variant we would expect to find if they are Anopheles coluzzii. For some of these individuals, you'll see that there is a region on the 2L chromosome arm that is coloured either blue or is yellow. So the blue is suggesting that it has a variant found in Anopheles gambi, and the yellow is suggesting that they're intermediate, heterozygote. But this is expected for Anopheles coluzzii, and that's because there is an integration that has occurred from Anopheles gambi at this region in the genome. And so we expect Anopheles coluzzii to sometimes have this profile. So we can classify species based on the fraction of aims that appear to be like Anopheles coluzzii. And we can visualize this on a plot up here. We can see that Anopheles coluzzii are coloured red at the top. They have a very high proportion of these aims that are considered to be as you would expect to find in Anopheles coluzzii. Down the bottom, we have our Anopheles gambi. Over here, we have a very high proportion of aims classed as Anopheles arabiensis using the other SNP set. And so those individuals are Anopheles arabiensis. And then we have a few individuals in the middle, so they couldn't be classified as any of these species. And these were the individuals that turned out to be Anopheles quadrangulatus. So these aims is using a large number of markers to identify species. So we can use them, but there are some drawbacks with this approach. One is that we cannot identify species that are not Anopheles gambiae, Anopheles coluzzii, or Anopheles arabiensis. And the other is that when these SNPs were discovered, it was based on a smaller set of samples from across Africa. So we may have more variation at these sites within these species than we might expect. And so in order to support this, we then did some further investigation. And that was using PCA and neighbor joining trees. So we performed a PCA, and this includes our Anopheles coluzzii samples and the rest of the samples that were Anopheles gambiae and Anopheles arabiensis and potentially an unknown species that we now know is Anopheles quadrangulatus, and compared these to samples from the AG1000G phase 3 resource, and also some samples from a literature dataset from Fontaine et al. And this includes some reference samples that we can compare ours to, including Anopheles merus, mellus, and Anopheles quadrangulatus. So the red individuals on this PCA plot are Anopheles coluzzii. You can see that all our individuals fall within the Anopheles coluzzii cluster. So they are like the other Anopheles coluzzii that we have within the resource. We have Anopheles gambiae in blue here. Again, all our Anopheles gambiae fall within a cluster with other Anopheles gambiae from across Africa. We have Anopheles arabiensis in green, and our individuals that we didn't know what they were, they fall within the reference samples of Anopheles quadrangulatus from Fontaine, and that's how we know it is this species. So to support this, we also perform neighbor joining trees. So these are a way to represent the minimum amount of evolution required to explain relationships among individuals. And this is done based on branch lengths. So basically, the closer individuals are in branch length, the more closely they are related. And again, we have the same color in here. So Anopheles coluzzii individuals are in red, and they form a very nice, distinct cluster with other Anopheles coluzzii from across Africa. Anopheles gambiae is in blue. We have arabiensis in green, and here's our Anopheles quadrangulatus. Of course, the finding that we have Anopheles coluzzii in Kenya is important because it may explain malaria transmission in Turkana. And so we wanted to then characterize this species further, and we did so by investigating their population structure. So we wanted to know how this population related to other Anopheles coluzzii. We used the same methods again, PCA neighbor joining trees, but this time we just restricted our samples to Anopheles coluzzii only, so we're not including the other taxa. So within the AG1000G phase 3 resource, we have populations from inland West Africa, Burkina Faso and Mali, coastal West Africa, Cote d'Ivoire and Ghana, and Central Africa, so Central African Republic, Cameroon and Angola. Our Kenyan samples are coloured in this dark blue here. And you can see that they cluster very closely with populations from inland West Africa. So from Burkina Faso, Mali, and also northern Cameroon. So Cameroon actually has population structure within it. Central Cameroon separates from northern Cameroon. So it's just northern Cameroon that's included within this cluster. And so what we have then is a finding that suggests that our Kenyan population is most genetically similar to inland West Africa and sort of northern Central Africa. We see the same thing when we look at the neighbor joining tree. Again, here is our samples here, and they are forming a cluster with the inland West African populations. So what this suggesting is that they are sort of genetically similar to these West African and Central African regions. And the fact that they cluster closely suggests that there is potentially ongoing gene flow. So we wanted to look at this further by looking at the measure of genetic differentiation, FST. So here we have a table of FST values, comparing all of our population cohorts, and Kenya is here. And if we look up at those values, we can see that the lowest values are present between the population in Burkina Faso and the population in Mali. The highest differentiation is with Angola in Central Africa. And then sort of the other values seem to sit between 0.01 and 0.017. And so certainly there seems to be very low FST between inland West Africa, suggesting that there is ongoing gene flow between these populations. We can also look at genetic diversity statistics to see how they compare to other populations. Here is Kenya. First of all, we can see that the nucleotide diversity values do not significantly differ from other populations. There's a couple of odd populations, which are Central Cameroon, Angola, which seem to have a positive to Gemins D, suggesting that they are bottlenecked. But in general, including populations in inland West Africa, values are similar. So this is suggesting there's no sort of strange demography that's going on. These populations don't show evidence of being an isolated population. And so they do have a profile that is similar to other Anopheles coluzzii. Another way to explore whether the Kenyan population is connected to West and Central Africa is to look at two regions on chromosome 2, which are known inversions that commonly occur in the Anopheles-Gambi complex. And these are the 2LA and 2RB inversions. So these are parts of the genome that have been sort of flipped. And basically, if you find that a population has the same variant or karyotype, that is evidence that there is gene flow between them. So we have our Kenyan population in blue, and we see that there's some individuals sitting in these two clusters. And this PCA is targeting just these inversion regions here. So we can see that the samples that fall with these are, again, the samples from Burkina Faso, Mali, and Northern Cameroon. And so they do have the same inversion karyotype as these populations as well. This is interesting because these inversions, the inverted forms, have been associated with arid environmental conditions. And all these locations that you see with this karyotype are arid regions. So it's interesting that they appear to be similar across different environmental conditions. So we also wanted then to investigate the molecular evidence for insecticidal resistance. We would expect a connected population to have similar insecticide resistance mechanisms. And of course, also this information is important for vector control. So what we have here, first of all, is amino acid substitution frequencies at the voltage-gated sodium channel gene. So this gene is the target site of prepheroids. And what we have in our Kenyan population is the L995F substitution. So this substitution is fairly well known. It's been associated with resistance to prepheroids, and it's at high frequency in coluzzii in Kenya at 62% frequency. And then we also have another pair of substitutions. We have I152-7T, and we have V402L. And you can see that they're at the same frequency. And it's been suggested that these actually do form a pair and they work together. And so that's maybe why you see them at the same frequency in this population. So recently, Mahamadi Kintega has reported this substitution pair from Burkina Faso. And interestingly, he's reported that it has been increasing in frequency across the years and actually appears to be going up while L995F is going down. So it is possibly replacing this sort of well-known substitution that is present across Africa. And interestingly, we see that it's actually, we have this actually quite commonly across Anopheles coluzzii. But certainly we see that both Kenya and inland West Africa populations do have this same double mutant. So in particular, it is concerning that this pair is present, and that is because it has also been shown that if a mosquito has the V402L substitution, this has actually a less of a fitness cost to the mosquito than the L995F mutation. And so it potentially gives peripheroid resistance, but at less cost to the mosquito. And because of this, it has the potential to promote strong peripheroid resistance that may reduce the effectiveness of PBO bed nets. So this is certainly concerning that we have this in Turkana. So we also looked at the GABA receptor, RDL, which is associated to resistance to dieldrin. And there are two substitution pairs of interest here. So we have T345M and A296G, and this is found commonly across sort of coastal and central Africa. The other pair, T45S and A296S, has actually only to date been reported in Burkina Faso, but we do also see this here in Mali and appears to be fixed in Cameroon Central. But interestingly, we see that in Kenya, even though it is at very low frequencies, there are some individuals with this substitution pair. So this is sort of, again, further evidence that they are perhaps have ongoing gene flow. And it may be that this proves to be more important and is on the increase in the future. So it'll be interesting to see what happens at these sites. So we also looked at acetylcholine esterase, and we were sort of interested in this G to ATS substitution, which is present here in Ghana and Côte d'Ivoire. But we see that that is not present in Kenya and Turkana, and it's also not present in the other inland West African populations as well. So we also looked at glutathione S-transferase, which is associated with metabolic resistance to peripheroids, but also to organophosphates and organochlorines. We see that in Kenya, we have the I114T substitution at very high frequency at 68%. And this particular substitution has been associated to resistance to DDT. And what we can see is actually it seems to be pretty common across Anopheles coluzzii, but certainly it is present in inland West Africa as well. So we found that Anopheles coluzzii appears to be a connected population, and it also is connected across ecological space. And that is because Turkana is a very dry, arid region in Kenya. And if we look back to this distribution map, Anopheles coluzzii seem to do well in arid regions, and this band actually extends across northern Central Africa into South Sudan. And so it's not inconceivable that this range would actually extend just a bit further into the arid region of Kenya. So it may be that this species was simply missed in previous sampling. And there have been sort of studies looking at species distributions or sort of the species that are present just based on routine entomological surveys that have been done in the region. But only Anopheles arabiensis has been reported. But previous studies have just used species-specific primers that differentiate Anopheles gambiae from Anopheles arabiensis, for example, but are not expecting to find Anopheles coluzzii, so they don't differentiate based on this. And so it may be simply that they missed if we look at previous sampling and studies, we can see that in one study, only 80% of samples reacted with a PCR band. And so, of course, it may be that there was low DNA quality or the PCR failed for some reason, but it may also be that these samples were in fact Anopheles coluzzii and they've just been missed in previous entomological surveys. We're not the only people to observe Anopheles coluzzii in East Africa. So recently, there was a report of Anopheles coluzzii around about here in Somalia. And so it may actually be that the range extends much further than was previously thought. Interestingly, both of the collections from Somalia and the collection in Turkana were from refugee camps where there's been very significant human development in recent years. And we know from the ecological differences between species that Anopheles coluzzii can survive in more polluted environments or environments modified by man. And so it does raise an interesting question because there's one scenario where the species has just been present in the region and has always been there. But another scenario is that it has moved in relation to urban development. So there is an interesting question that is raised here. Of course, the presence of Anopheles coluzzii in Turkana does have consequences for malaria transmission, and more needs to be done to understand its role. Previously, only Anopheles arabiensis was thought to be causing malaria in the region, and this vector was considered less competent. And so it may be that actually there is a higher malaria risk than expected. The study, finding a species that was unexpected, highlights how genomics is a useful tool in taxon classification, and it is perhaps a more comprehensive and accurate way to identify species and species ranges. We found evidence that the population of Anopheles coluzzii in Kenya likely forms a connected population across inland West and Central Africa. So if there are insecticide resistance variants that arise in a connected population, we expect those substitutions to spread via gene flow to other geographical regions. So this highlights the need for a coordinated effort across country boundaries. We found that the Anopheles coluzzii population had genomic evidence for insecticide resistance, and so malaria control in Turkana needs to consider these mechanisms when deciding the best approach to use in the region. It looks like there is resistance to prophylloids and DDTs, and so methods that can reduce this impact would be preferred. Our study demonstrates that whole genome sequencing allows lots of useful information to be acquired at the same time. So we get information on the species present, but also insecticide resistance mechanisms. And so implementing this on a routine basis would allow us to assess how populations respond to control over time. And it's a useful tool that could be applied on a routine basis. Finally, our study also highlights the fact that we don't know the full extent of the Anopheles coluzzii range. And since this is a major malaria vector, this signals that there is a massive gap in our knowledge. And so more comprehensive sampling certainly needs to be done, preferentially with some whole genome sequencing to see if these really are Anopheles coluzzii. But certainly we need to understand whether the species actually has a much it has an extension of its range across the African continent. So that brings me to the end of the presentation, which leaves me just to thank everyone involved, everyone authored on the paper, but in particular, I'd like to thank Luna Kamau, Sonia Barassa, Alistair Miles, Jon Brennes, and the Genomic Surveillance Vector team. So thank you very much for listening. I'll hand back over to Babalo.
Thank you so much, Kelly. This was amazing. Well explained, very patient, and yeah, easy to follow. So thank you so much for your time and presentation. It was brilliant. We are now have reached time for Q&A. We have about 18 minutes to go. So kindly raise your hand and we will select you to ask your question. We'll unmute you, or you can type your question in the Q&A box or the chat box, and Kelly will be able to engage with you. But while people are thinking or raising their hands, I have a few questions of my own, but I think I'll just ask one. So I think in your second slide, also you explained how Anopheles coluzzii is mostly found in West and Central Africa and hadn't been sort of seen or evidenced in East Africa, particularly Kenya. And I think at the end you did explain that there is some emerging evidence that it's showing up in Somalia as well, and the reasons for that. But my curiosity is, are you thinking or saying that Anopheles coluzzii has always been present in Kenya, however it wasn't detected, or that something is changing in terms of climate change, the environment, and other factors that are making coluzzii move into East Africa and other parts of Africa that it wasn't previously present in? Kelly?
Well, certainly we can say that it's been present since 2006, as that's when our first sampling was. But yeah, I think it's an open question. It would make sense given that it's sort of found across similar environmental conditions to the distribution that we actually know about, that it actually just would form an extension of that distribution, and it's just been sort of missed off based on sort of standard diagnostic techniques. So that would certainly make sense. And of course, we see that there is evidence for gene flow. So we again suggest that there is sort of an environmental corridor where this species is sort of able to exist, and that has presumably been the case for a long time. But because this vector is associated with more urban developed regions, and there has been a lot of development in these regions that were once sort of more, I guess, traditional sort of farming lands. There is the question of whether this species was present or has moved in with in urban development. But I think that is something that we unfortunately cannot answer with our data at the moment. But we now have haplotype data, which we didn't have before. And so this is something that could possibly be looked at in the future.
Thank you so much, Kelly. There's a comment from Jane Mwangi. Really nice talk. Thanks, Kelly. That's from Jane. As I said, we've now reached our time for Q&A. So please either raise your hand, we will select you to speak, or type in the Q&A or chat box, and I'll read out the question for Kelly. Okay, the second and hopefully last one from me. So the 20th of August is an annual day that we celebrate Mosquito Day, World Mosquito Day. And one of the hashtags or messages that are going around is zero malaria or end malaria in our lifetime. Because malaria is one of the, particularly for Africa, has a huge impact on Africa, and mosquitoes is the world's deadliest animal. Do you think with what you've discovered in this paper on this research, will we be able to achieve zero malaria in our lifetime, or do we still have longer to go with the introduction of new insecticides resistance, etc.?
I would argue that just because we've seen a reduction in cases, that people shouldn't take their eye off the ball. And yeah, certainly I think there's a lot more work that has to be done. We've made gains, but people shouldn't sort of forget about malaria. As long as people continue to put pressure on malaria, then yes, I think we can eliminate malaria within our lifetimes if sort of, I don't know, the funding shifts away, or people sort of don't kind of continue with the same drive or passion, then perhaps not. But I think certainly it's achievable, and that we've made amazing gains so far.
Thank you, Kelly. Hopefully people don't take off the pressure and continue fighting malaria. I'm checking the Q&A box and the chat to see if there are any questions trickling in. I'm gonna give it a few minutes. As I said, you can raise your hand, or you can type in the Q&A or chat box. And as I said, Malaria Gen and GSU ran a nice campaign on the 20th of August. Please do go through the hashtag World Mosquito Day. You will find resources on verbs and training and data and other resources that has to do with the work that we do at Malaria Gen. And I think it's quite educational and could be beneficial to your work as well. So please do check it out in your spare time. Okay. If there are no more questions or comments, I'm going to ask Kelly if you have any last words before I close today's session.
No, simply that thank you so much for allowing me to talk today, and I've loved it, and you've been a great organizer. So thank you.
Thank you so much, Kelly. You're a great presenter and speaker, very knowledgeable and accessible as well. So thank you so much for your time. To all of you attendees, thank you so much for attending today's Jenna Club. There's a comment from Ishla O'Connor. Thanks so much for such a wonderful presentation, Kelly. And I just want to repeat that as well, just to say this was a wonderful and great presentation, Kelly. So thank you so much. To all of you attendees, we really appreciate your attendance. We are going to have another Jenna Club on the 25th of September. So kindly be on the lookout for adverts on our social media and website and newsletter for that as well. And you can register on time. Sonia Barasa says, great talk, Kelly. Congratulations. And I just want to agree again with Sonia. And for now, I'm just going to close today's Jenna Club and say there's going to be a recording online. Check out our Malaria Gen YouTube channel later today. The recording will be online and you can share with those that couldn't attend today. Thank you so much and enjoy the rest of your day. See you next time.